Okay, I'm pleased to introduce today's speaker, Dr. Matthew Holding, who comes to us from Clemson University, where he's currently a postdoc working with Chris Parkinson, and he's about to start a new postdoc at Florida State University for 2 years with Darren Rokita. Before that, he did his PhD at Ohio State with Lyle Gibbs. And I guess you interacted with Andreas Chavez while you were there, so a recent postdoc from here, who probably many of you know. And before that, did a master's at Cal Poly with Emily Taylor, also well known here. And he did his undergraduate work at Ball State University in Indiana, where he said he didn't get enough training in basketball or something. So we ask, well, so Matt's work has explored the coevolution between rattlesnake venoms and squirrels on which they prey. And he has a paper with the interesting title, Snakes Win, and then more about coevolution. So I hope we're going to hear something about that. As a mammologist, I don't like that title. We asked our speakers to provide some fun facts, and he provided some very fun ones. He said, I decided to become a biologist after David Attenborough wrote a reply to a letter my mother wrote him when I was 7 years old, telling him how much I loved his documentaries. So I guess David Attenborough.
We were listening to Planet Earth in the lab down there the other day.
Oh. And he was also invited to be a cast member of Venom Hunters. And he's happy that he was advised to turn down the offer. I'm ashamed to say I don't know what Venom Hunters is, but I guess he did the right thing. So please join me in welcoming Matt Holding.
Hi everyone, I'm happy to see all of you today. First I want to thank everyone here at the MVZ for making me feel really welcomed and kind of part of the team over the past week. I've been measuring some snakes as part of my use of collections postdoc fellowship that Michael just mentioned. I also want to thank you, Michael, for extending the invitation to me. And by extension, I should also mention that I want to thank Andreas Chavez, who became both a colleague and a friend when he came to Ohio State, and who is the person who originally recommended me for a speakership spot here at the MVZ lunch. As was mentioned, I'm currently kind of a joint postdoc between Clemson University and Florida State University. But what I'm going to talk about today is the research that I completed back in the previous May, last May, which led to me earning my PhD in Lyle Gibbs' lab at Ohio State University in the Department of Evolution, Ecology, and Organismal Biology. I'm going to focus mostly on these two organisms, the northern Pacific rattlesnake and the California ground squirrel, which are a common animal that I'm guessing the vast majority of folks here in this room have encountered on some hike or trip through their backyard or another here in California. But more broadly, I'm interested in interactions between predators and their prey. These interactions are important for us to study because of their ubiquity. Nearly everything is eaten by something else, and so our understanding of the maintenance and the generation of phenotypic diversity in nature is going to hinge in large part on our understanding of the evolutionary factors that influence the outcomes of predation. Do we have a successful offense or successful defense of the predator or prey respectively? Oops. When we think about predator-prey coevolution, the idea of an arms race often comes to mind, and this is another system that's been broadly looked at across the state of California, that of toxic newts that are fed on by resistant Thamnophis garter snakes. And back in the early 2000s, Brody et al. Showed this kind of nice pattern that sums up the idea of an escalatory coevolutionary arms race, where we have traits like the resistance of the snakes being positively correlated over several spatial locations with the toxicity of the newts. Even for a classic representative system like this, things can often get complex, and it turns out that in this system, when more sites were added, that nice positive correlation breaks down, particularly at the extreme values of garter snake resistance. The broader takeaway from results like this is that coevolution between predators and prey and species interactions in general are highly context-dependent processes. And so homing in on those contexts and understanding the rules of how they govern the successful predation or defense of predators and prey is going to be what is important to our understanding of the complexities of biological diversity, and that's specifically what I'm going to be focusing on here, drilling down and understanding, picking apart some of those concepts. Most of that work to date in empirical systems has been done in host parasite systems. These are systems that are much easier to study often. You can plant experimental plots of some plant of interest and sprinkle different genotypes of fungi in it or infect it with guilds of choice of particular herbivore taxa. I'm going to argue today that venom-mediated systems, such as that between snakes and the resistant mammals or other prey that they eat, offer some of the same types of benefits as the host parasite systems so that we can begin applying coevolutionary theory as it's currently understood in mathematical models to predators and prey and not just to hosts and parasites. And that's because we can do a lot of work in vitro. The venom and prey tissue can be actually lifted off of the organism by things like a venom extraction. And then we can do in vitro tests that give us some idea of functional resistance of prey to the venom of interest. Many animals across the tree of life have evolved resistance to the venomous taxa with which they interact. These include eels that are eaten by sea snakes, all the way to other snakes that actually feed on venomous snakes. And there are a whole slew of mammals that have been shown to be resistant to venoms of the snakes that they interact with. As I mentioned before, these are going to be the stars of today's talk, the northern Pacific rattlesnake and the California ground squirrel. This system fits the mold for coevolution and became the marquee system that I chose to study this snake-prey interaction within because there's a large body of work that suggests that this system fits the mold for a close, ecologically and evolutionarily important coevolutionary dynamic. First, Henry Fitch, working partly here and with other organizations in California way back in the 40s, kind of started this work where he showed that at the San Joaquin Experimental Range in the Sierra foothills, up to 69% of snake diet by weight can be composed of just ground squirrels. So this is an important close interaction. The squirrels deal with this high threat of snake predation with first a diverse suite of anti-snake behaviors. And these have been well studied by the lab of Dick Koss at UC Davis for quite a number of years. So for example, this is a rattlesnake's shed skin right here. And this squirrel has chewed it up into a paste and is applying it to its fur. This has been shown to give it a chemical masking. The paper was entitled "Donning Your Enemy's Cloak." And so rattlesnakes don't like squirrels as much when they smell like another rattlesnake. You can see why that might be confusing. The most famous of their behaviors is tail flagging. I've actually used this behavior to find rattlesnakes in the field. If you see a big group of squirrels that are all waving their tails and chirping, there's usually a rattlesnake at the middle of that circle of squirrels. This has been shown recently by some researchers at San Diego State to be an honest signal of vigilance. The squirrels are saying, "I see you, go away," and the rattlesnakes usually choose another hunting location. The squirrels back these behaviors up, which are somewhat risky behaviors, they're getting right in the snake's face, with venom resistance, particularly to venom metalloproteinases. If you see nasty pictures of snake bite where a finger is dropped off or there's rotting tissue, it's these venom metalloproteinases that are to blame. They destroy tissue, they open blood vessels, cause swelling, and allow other venom proteins to dissolve out to do their killing work. They're thought of as gateway toxins in that regard. The squirrels have serum inhibitors that are constitutively expressed in their blood and will irreversibly bind and inactivate the venom of these snakes. This resistance was shown to vary geographically across the state of California and into Oregon, where the species are interacting. However, the venom used in those studies was obtained from a single stock source. It was purchased from a company like Sigma. And so one opportunity that we saw was to bring a parallel study across the same geographic range of snake venom and ground squirrel serum resistance to try to understand whether local adaptation and coevolution might be playing a role in the evolution of this system. So just a couple more anecdotes from some of the early important work on this. This is a squirrel from a resistant population and a squirrel from a non-resistant population that were injected with the same venom in the same concentration. You see the hemorrhage caused by the venom metalloproteinases here, but the resistant squirrel is going to survive. It's only got a tiny little patch of wound. This one died after massive hemorrhage to that rear quarters. And finally, I was able to mentor a couple of undergraduate students to show recently that at least at the inter-specific level, there seems to be adaptation of the squirrels to dealing with their local snake predator. So we collected serum from ground squirrels out here and venom from rattlesnakes, and did the same thing with the local tree squirrels that live in Ohio and the local timber rattlesnakes that feed heavily on those tree squirrels, and showed the following general pattern using what's called a serum inhibition assay. And I'll show that in a minute, but basically the gray squirrel was best at inhibiting the timber rattlesnake, here in orange, whereas the California ground squirrel was best at inhibiting the northern Pacific rattlesnake compared with the timber rattlesnake venom. So we see this crossing pattern in the graph that suggests that at the species level, these squirrels are adapted to dealing with the snakes that are feeding on them. This makes a lot of sense. Today, I'm going to focus at the population level across the state of California to test 3 ideas about basically contextual factors that could be influencing the coevolutionary dynamics of snake-squirrel interactions across several California sites. First, we're going to test for the presence of local adaptation, and then go on to use it to infer the form of coevolution occurring in this system. Do we have an arms race, an escalation, or some other type of interaction? Second, we're going to test the idea that the evolutionary potential of snakes and squirrels at each of our populations is limited by genetic demographic factors. And finally, we're going to bring additional context into this story by thinking a little bit about the other prey that these snakes eat. They feed a lot on squirrels, up to 69% of their diet, according to Fitch, but they eat a lot of other things as well, and we can't forget about that. So let's start first with just a test of venom or serum local adaptation in this system. How would a test of local adaptation look? To give it in a basic sort of cartoon sketch here, if we had 2 populations of snakes and squirrels, maybe 2 locations, blue and orange, we could test sympatric versus allopatric combinations of venom and prey tissue and do a contrast. Do sympatric or allopatric patterns show higher or lower functional outcomes of envenomation? And then what is the order of that pattern? Is it the snake or the squirrel that's performing best locally versus in a foreign environment? Another way of looking at this, if we had some mock data here, say we had a measure of venom performance in the presence of our prey, and we injected venoms from 2 sites into prey 1, prey population 1, we might expect that snakes from that same population would outperform snakes from a second site. Oops, sorry. But if then we looked at the site number 2, we would see this crossing pattern, where we expect that snakes from that local site would outperform the foreign snakes. You can think of this as a spatially segregated antagonistic pleiotropy or a trade-off model, but basically what it is showing is local adaptation, in this case, of the rattlesnake. The snakes perform best with their local squirrels. An X like this flipped on its head would represent squirrel local adaptation. So this is just to give you an idea of what to expect as I go through the actual results of this experiment. Furthermore, if local adaptation is present, it actually tells us something about the nature of putative coevolution in this system. And let me tell you why. In those escalatory arms race dynamics, the pattern we expect, the prediction under this arms race hypothesis, would be for this positive correlation between trait means to exist. Do we see toxicity and resistance positively correlated over space? These types of interactions are about differences. Who's more toxic, who's more resistant. There are another type of interactions that can decide the outcome of predator-prey meeting in nature, and that is a match-based or trade-off model, where instead of about who has more, it's about the fit of one trait to the other. How well do the inhibitors of a squirrel show a molecular match to be able to quickly track down and inhibit the venom once it's injected? It's only under these types of matching-based models, where we don't expect this escalation to occur, that we would expect to see this reciprocal trade-off across space, where local versus foreign contrasts lead to a pattern of local adaptation. So we collected about 300 venom samples from across the state of California back in 2013, and at the same sites we collected blood samples from ground squirrels and used this to gather serum, which contains the venom inhibitors of interest. So at these 12 sites we were able to gather at least 10 adults of each species per site, and then each snake's venom was tested with 12 squirrel serum samples in a serum inhibition assay. One sympatric or local squirrel, and then one squirrel from each of the 11 allopatric or foreign sites. And this resulted in 1,440 unique snake-squirrel combinations in the data set that I'm about to present across these 12 locations. In the lab, this is a pretty simple assay. We essentially are going to dilute to a standard concentration our snake venom and our squirrel serum, and then incubate the venom in the serum pool for a standard time prior to measuring the metalloproteinase activity of the snake venom. We can do this in the presence or absence of serum, and then calculate a change in venom activity. So if serum is present, the venom is going to be less active. That's a ubiquitous fact in these assays, but the amount of activity lost by the venom is going to give us a measure of how resistant that squirrel was. And some of the early resistance out of early work out of Dick Koss's lab at UC Davis showed a strong correlation between actual measured lethality of these snake venoms and the ability of the serums to bind to snake venom in vitro. And so we're going to use those two measures to get a change in activity score, as I just described. Venom plus serum minus venom only gives us a resistance functional measure of the snake-squirrel interaction. So first, do we see escalation evidence? Does our prediction of possible positive correlations over space between venom and resistance hold up to support the arms race? And the answer is no. So we see no evidence of a relationship between these traits. What about this idea of matching? And to evaluate that hypothesis, we need to actually look for whether local adaptation occurs in a sympatric versus allopatric contrast of all of these different measurements. Before I talk about that, I'll say what actually did predict our venom activity and our squirrel resistance over space. It wasn't the trait of the enemy species. Instead, it was the site elevation of the local sites. We had a strong positive relationship between site elevation and venom activity in the snakes, and a weaker but still significant negative relationship between site elevation and squirrel resistance. Now, I think there's some fascinating physiological work to be done to actually understand the proximate reasons why do we see these relationships, and I can talk about that at length in terms of our ideas. But for me and my purposes of detecting local adaptation, what this represents is some sort of noise that we have to control for. And we did that by splitting our sites into 6 high and 6 low elevation sites to control for elevation in our model of local adaptation. And then we developed the following linear mixed model to test for local adaptation in this system. Now, I know everyone gets really excited about going through a big ANOVA table, but I think it's important to do here to tell you what this model looked like. We had main effects of the elevation of origin of both the snake and the squirrel sample in a given snake-squirrel test, the population of origin as well to control for geographic main effects of where the snake or squirrel samples were taken from. And finally, the individual identification numbers of the snake and squirrel were used to control for repeated measures of each venom sample and each squirrel blood sample across the 12 tests that it was used in. And finally, we had our local adaptation term, and this is the key. Was the given serum venom comparison a sympatric or local comparison? Was it an allopatric comparison across the same environment type? Or was it an allopatric comparison from one environment type to another? Was it a high elevation squirrel with a low elevation snake, for example? And all of these terms were statistically significant predictors of our snake-squirrel pairings, or the change in activity score, and importantly, the local adaptation term is significant. And so here's a post hoc analysis to understand the direction of that local adaptation and exactly how it's occurring and by how much. So here's that change in venom activity score. Lower is better for the squirrel because it's taking more venom activity away from the snake. The sympatric comparisons and allopatric comparisons from the same environment type show responses that do not differ significantly from one another and are almost exactly the same, actually, across these different comparisons. But the allopatric different elevation scores show a reduction, a further reduction in venom activity when serum is present. In other words, the snakes perform 10% more poorly when they are challenged with the inhibitors of a squirrel derived from a site that is away from home and very different in environment. In other words, the snakes are locally adapted to ground squirrel venom resistance with an overlay of an environmental factor structuring that local adaptation. Finally, we can look at this to try to derive our crossing pattern that I set you up to expect. If we just categorize each venom and serum pairing by whether it was from a high or low elevation site. And we see that. Although there's a general trend towards more resistant squirrels at low elevation, high elevation squirrels are best attacked by high elevation snakes, whereas at low elevation, the low elevation snakes perform best. So, conclusions from this first part. First is that population variation in snake venom may not only be driven by differences in the broad diet of the snakes. So previous studies have focused heavily on the diet composition of snakes. Maybe snakes over here eat a lot of lizards. Over here, we're in another population, a lot of mammals are consumed. Instead, we now have evidence to suggest that both this kind of broad diet composition, but also variation in the resistance phenotypes of single prey species may be an important driver of, and maintainer of venom diversity at the population level. All squirrels, from an evolutionary perspective, are not created equally in terms of their ability to select for and maintain snake venom diversity. This leads to an idea that we could have this matching-based interaction. We don't find support for escalation, higher toxicity, higher resistance. Instead, under these matching models, there could be a gene-for-gene effect. A collaborator of ours has just done a genome for a rattlesnake, and they found 25 copies of snake venom metalloproteinases in the one genome. So if each of those genes is potentially responsible for killing a certain prey or avoiding a certain prey resistance factor, you can imagine scenarios like this where we get these local adaptation patterns. Certain populations of prey have certain inhibitor suites in their serum, and venom in turn is selected to diverge across those populations accordingly. Finally, it was surprising to us initially that the predator was the locally adapted species in this interaction. Ideas like the life dinner principle would suggest that prey, which are going to die if they lose in one of these interactions, have a stronger fitness drive to be the winner in an interaction. So what we wanted to understand was how is it that the snakes are the locally adapted organism in this interaction. And to do that, we're going to break out this broad summary of a pattern into individual populations. And so this is what that looks like. This is another way of visualizing that change in venom activity. And basically, snakes are consistent, the pattern is consistent with snake local adaptation when we have a positive value of this score. But if we have a negative value of this score, that's more consistent with a population where the squirrels are possibly the locally adapted player in this interaction. And so we see 4 of those. So it's interesting to think about why what might be different about these 4 populations. But more generally, we have a lot of interesting variation here that we can potentially leverage to test for other ideas about those contextual factors that I predicted to be important in local interactions between predator and prey. And in particular, I'm going to focus on genetic demography, which has been heavily considered as an important factor influencing predator-prey interactions and coevolutionary actions in general. So a lot of this work comes from mathematical modeling that has been done by Scott Newsmer and Sylvain Gandon and their colleagues. And 2 demographic factors have been hypothesized and shown to be important in deciding the local evolutionary potential of a co-evolving enemy. First is effective population size. If you have a larger effective population size than your enemy, this is a good thing, and that's for 2 reasons. First is that there's likely to be more standing variation around in the first place, upon which selection can act to allow you to get an evolutionary edge over a co-evolving species. And second, and maybe a little bit less appreciated, is that there's a lower power of genetic drift in these large effective size populations to move populations off of that local adaptive fitness peak. Second is migration. Under some circumstances, it can be good to have more migration than your enemy. And so more migration can actually be a facilitator of local adaptation. This kind of goes against the idea of migration as a homogenizing force that brings populations together in genotypic space. However, under some circumstances, migration can introduce variation that's been previously lost to genetic drift. That would look like this, right? This is a context-dependent process in and of itself. So some nice work by Jeremy Yoder and Scott Newsmer actually allows us to infer sort of a decision tree for how effective population size should affect our predictions about how migration might influence the local coevolutionary potential of predator and prey. In particular, if we have really small effective population sizes, these are the instances when genetic drift is likely to be occasionally pulling variation out of that population and allowing for that genetic rescue factor to come into play in the first place. Otherwise, in large populations, we don't expect that to occur in the first place. And instead, migration is just going to introduce potentially maladapted individuals that pull the population off of its local fitness peak. So under under large effective sizes with maybe a key value of 1 migrant per generation, we expect migration to be a maladaptive force. But if we have small effective size populations, small numbers of migrants per generation, we expect a larger migration rate to actually be a potential force for rescue and an increase in local adaptive potential of predators and prey. So for this work, we did some comparative population genetics using RADseq, and we focused only on our southern populations because in the intervening time between when I made my initial collections and when we did this work, a nice study from Craig Moritz and a student of his here, Mark Fung, came out that suggested that the northern squirrels are actually pretty extensively diverged from the southern squirrels, and they actually suggest elevating those to a different species, Otospermophilus douglasi. And so we wanted to focus on a fairly well-connected metapopulation, and so we have these 7 southern sites from which we collected DNA samples from the snakes and squirrels, and got about 1,000 RADseq loci from around 70 individuals of each species, about 10 per site. We tested the following demographic models in the program FastSymCol 2 to understand connectivity between these populations, and this full model of stepping stone connectivity between the populations was our best supported model. And then we used this model to estimate those demographic parameters of interest, namely number of migrants per generation and effective population sizes of each of these local populations. So we have our full model here. Each of these black bars represents migration. And so, for example, for these three populations, for each location we would have the effective population size estimate as well as bidirectional migration rates for each site. And so what is that genetic demographic background that we need to even make informed predictions about how migration should influence this predator-prey interaction? So another surprise from the rattlesnakes was that they tended to have larger effective population sizes than the ground squirrels. So this initially gives us an idea that the snakes might have sort of a demographic edge from the effective population size point of view. And also, all of the effective sizes of both the snakes and the squirrels, this is the number of migrants per generation, were greater than this sort of critical value of 1. It takes us down this side of our decision tree, where we're going to predict that a larger NE than your enemy is a good thing, but that migration might be a maladaptive force for local adaptation in this system. Because we have a comparison between our rattlesnake and our squirrel at each local site, we can then calculate a simple difference to give us an idea of how these values compare across each of our geographic sampling localities. And that was a simple snake minus squirrel NE, as well as M value for migration. Under hypothesis 1, that larger effective population sizes facilitate local adaptive potential, this difference in NE value, is predicted to be positively correlated with our local adaptation scores. That bar graph that I showed that broke each population out one by one from our serum Venom comparisons. And under migration, because now we're expecting it to be a maladaptive force, we'd expect the opposite, a negative relationship between migration rate differences between the snake and the squirrel and local adaptation in the system. Okay, so what did we find? With regarding to effective population size, we show that there's a moderately strong and significant association between differences in effective population size of snake versus squirrel and the local adaptation score for that particular site among these 7 sites of interest. So to put this into sort of a biological context, the 4 sites where snakes had larger effective population sizes than ground squirrels were the 4 most locally adapted snake sites, whereas these 3 sites where the squirrels had larger population sizes than the snakes, according to our estimates, were the least locally adapted snakes. We see the expected negative slope with regard to our migration estimate. However, this is a non-significant result. So we'll move on from that. All right, so conclusions from this section. So we see that a larger local effective population size is one of those contributing factors that gives us some context to explain variation in this co-evolving interaction over space and possibly over time. And it also tells us why we get this initially surprising result that the snake, the predator, is the locally adapted species. Part of that, at least, might be that their effective population sizes were larger. Now you might be thinking, how can a predator have a larger population size than a prey? It's important to remember that these are effective sizes and therefore can be correlated with census size but aren't necessarily so. These squirrels are more habitat specialists. They're living in particular soils that they can build their burrows in, and they also aren't as vagile as the snakes. So there might be more connectivity between the snakes, keeping the genetic diversity high in these populations. All right. Finally, here we see that migration, which is usually talked about in the host parasite literature as an adaptive force, and usually we're looking at, say, a host that has a higher migration rate than a parasite tends to be the locally adapted species. However, it's important to remember that, at least from our mathematical modeling perspective, there's sort of an additional layer of context to this factor in terms of how we would actually expect it to be predictive. And I hope that our work here is sort of a demonstration of how that can be considered in an appropriate context. All right, so lastly, we're going to bring in some other prey species, because although these snakes eat a lot of ground squirrels, they eat a lot of other things as well. Mammal and bird people, part of the vertebrate group. I'm sorry, I'm just showing a lot of dead vertebrates. I think this is our first bird. It is a European starling though. So, how does this broad diet that these rattlesnakes consume possibly influence the evolution of their venoms? What we did was, from the same snakes that we collected venom from previously, we generated HPLC profiles of their venom that give us a relative abundance measure across all of the different toxins that are present. This is an HPLC profile. Each of these peaks represents one or more venom proteins that are in the venom of a single snake, and the height of those peaks is proportional to how much of that particular protein is in the venom. We summarize that data with several different multivariate analyses, and so with something like a PCA, we can see major geographic splits in average venom phenotypes. There's a north-south split that's kind of concurrent with the Bay Area, as many other biogeographic data show, whether it's phylogeography or, in this case, we're talking about a trait that's split across the Bay. But there's, at each, if we look at population grouping, we see that although this is a little bit of a mess, there's kind of each population occupying its own little part of the venom phenotype space. So this is a cool pattern to look at, but can we actually explain it with anything that we believe to be relevant to venom evolution? We used a diet study that reviewed several rattlesnake diet studies, so this is kind of a diet meta-analysis, and combined that with the California Wildlife Habitat Relationships Project, which provides fairly detailed range maps for several small mammal taxa. And so we identified 44 potential prey and simply used range map overlap to call something as present or absent at a site. So we have two sites here. This site would have both of these mammals present. This site we mark only one as present, the other is absent. I know this is very coarse, but it turns out we actually learned something interesting, suggesting that even at these coarse scales, prey community has some interesting predictive power. Just to look at the diversity of the prey that we included, 5 shrews and a couple rabbits, and then several rodents. We included our lovely, majestic California ground squirrel, and we also had another rodent, several neotoma wood rats. They're also known to be highly resistant to snake venom, but with a slightly different mechanism. And then a bunch of other smaller rodents that are heavily present in the diet of these rattlesnakes, like kangaroo rats. In 2017, a paper was published that studied the same geographic area that I did. Each of these black dots is a venom sample. They showed that the abiotic environment, these were world climb bioclin variables, predicted venom variation in HPLC proteomic profiles. So this kind of provides an alternative to the prey community. Is the abiotic environment important? Or if we actually add in prey data, does that give us an even better predictive power in terms of understanding venom variation over space? Maybe the abiotic environment is just a surrogate for the prey that are present at a given site. So we summarized first our genetic data to control for genetic distance between sites with ordination. We summarized our prey community data. We summarized abiotic environmental data and tried to predict our complex venom phenotypes using these different markers of variation at each of our 12 localities. Turns out that the prey community axes are pretty highly correlated with the environmental abiotic environmental axes. This makes sense. If it's colder or wetter, you tend to get a different suite of mammal taxa present. So we're going to control for genetic divergence and then test the ability of the abiotic environment versus the prey community as predictors of venom composition in these populations of snakes. Here's what this looked like. So this is a redundancy analysis. I won't go into the details, but basically each of these is a site. The size of these circles represents the number of vole species that I marked as present at each site. And the color that I'm now adding to these marks the number of kangaroo rat species that are present at each site. I've kind of skipped the punchline here, which is that the prey predicted significantly 23% of the variation in our venom phenotype across 130 venom samples. And although the abiotic environment was a significant predictor, we see that it's not as effective a predictor, suggesting that the abiotic environment was probably a surrogate for which prey are present. Back to the meat of the discussion here, we kind of can infer in these data a sort of wet to dry, and voles are sort of more meadow, wet area species, to dry kangaroo rat diverse habitats axis across these sites. And so it's possible that voles versus kangaroo rats, in addition to variation in resistant ground squirrels, which were present in all these sites, has a selective influence on the venom phenotype in these snakes. Okay, so when we see different, very different venom phenotypes over space, it's possible that the particular prey species present is another important selective factor for this. And this has been shown previously in other work, but not necessarily with data just from which prey are present or absent. These were diet studies. Those diet studies can actually be biased because if a snake eats a large prey item like a squirrel, it's not likely to move around and be active on the surface, and therefore it's not likely to be captured by someone that is collecting for museum accessions or picking animals off the road. So in this way, we can use prey community information to sort of circumvent the diet studies, or at least supplement the diet studies, as a way to actually predict functional variation in the venom phenotype, in the venom composition of these rattlesnakes. This sets up some interesting hypotheses that we could test, right? You could take venom samples that I have from vole-heavy areas versus kangaroo rat-heavy areas and do lethality tests or other types of tests to actually add further support to the idea that you might have kangaroo rat versus vole-specific venom phenotypes out in the environment. And finally, because of the combination of the work that I've done here, we see that in the same venom, you have two possibly competing selective forces, or maybe complementary selective forces, acting on this complex phenotype of venom. You have differentially resistant ground squirrels, so one species that's really important, and then superimposed on that, we have other important prey that these snakes feed on that maybe have just basally different physiologies that the venom is selected to kill. Venom is a very complex trait, so another option, right, is that this is sort of a toolbox, and you have a wrench for ground squirrels and a hammer for voles. Generally, I think to come back to where we started with maybe a little bit more of a change from where we started is an emphasis on the idea that the outcomes of predator-prey interactions over space and time are highly context-dependent processes. And factors like the genetic demography of local populations and the mode of coevolution that is mediating the predator-prey interaction are going to be the key forces that we need to understand in order to predict and describe the variation present that we observe in nature when we do things like make museum collections or collect protein samples, things like that. And so in this case, we saw that the types of traits involved, the specific prey species, and the resistance of certain prey species, and the complex environment with different elevations and different temperatures is going to all come together to produce the traits that we observe when we do a local population study. More generally, I want to talk about a few future directions from this that I have outlined for this work. One is to characterize the specific proteins that are involved. So we know that metalloproteinases and their inhibitors are important, but what are they? The student that has taken up the helm in my place in Lyle Gibbs' lab is co-advised by Andreas Chavez. And what we've done so far is to use kind of a bait capture approach where we fractionate the venom and then see which serum proteins stick to each venom protein. And Andreas is going to come in and help with some transcriptomics, where we're then going to look at expression variation of each of the genes we identify as being important at the molecular interface of the snake-squirrel interaction. We can ask these same questions in different species. So these northern Pacific rattlesnakes that I studied here have these large, highly connected populations. Some of the sites we were able to collect over 20 snakes in less than 6 hours. Other rattlesnake species, other viper species like eastern Massasauga rattlesnakes, live in small, highly fragmented populations out in the agricultural desert of Ohio and Indiana. The effective population sizes of these animals can be as low as 10. These might be the situations where migration would be predicted to be a maladaptive force, and if you can get a little bit of connectivity, you get some genetic rescue that leads to higher adaptive potential to deal with evolving resistant prey. And finally, we can ask about the drivers of venom diversity from populations all the way up to species. And that's actually what my postdoc that I'm now working on is trying to do. Applying some of these population level ideas as well as other approaches, I'll just give a quick plug on that. So this is a photo that I took yesterday down in the other corner of the museum. We're getting measurements of morphometrics of the venom delivery system. And the 9 months prior to the beginning of this month, I was a postdoc generating proteomic and transcriptome data for several pitviper species. In green here is our coverage of the rattlesnakes, cottonmouths, and copperheads that consumed 4 months of my life sitting under a hood at a lab bench generating those transcriptome data. And so we're going to combine morphology and our information from these molecular genomic techniques to try to understand co-adaptation of the venom delivery system and the venom phenotype at the molecular level over a large biological. With that, I want to thank you all for your attention. I have to mention my advisor, Lyle Gibbs, and my lab, several of the UC reserves and other Cal state lands and federal lands were really helpful in this project. And I want to mention my funding sources. I was funded as a student at Ohio State by an NSF graduate research fellowship. All of the actual research involved in this was from small grants of less than $1,500. So I'm really, really thankful for the existence of those resources that allowed this project to be done. Thank you much.
All right, we have time for questions.
Yeah, great talk.
Thank you.
One quick question about, I know it's already very complicated, but you mentioned with the elevation gradient, there were differences in resistance and toxicity. And of course, you prefaced the whole thing by there being behavioral resistance as well. So it seems like an obvious possibility is that the behavioral resistances are different to different elevational gradients, and therefore changing the selection on antivenom.
You're thinking a trade-off in strategy.
Sure.
I haven't thought about that at all. That is, I think, a great idea. We know from the original work that was done with behavior that they did geographically structured surveys of behavioral intensity. How intense do the squirrels react when a rattlesnake is tethered near their burrow? And there was a lot of variation in that. So that exists, and we could look for those types of trade-offs.
I'm thinking of the results from the first part of the talk. Has anyone looked at whether there's any plasticity in the snakes and the strength of their venom? Because I think one explanation could be that they're able to sense somehow the resistance in their prey or what elevation they're at, and then, maybe if you're a young snake and you eat a highly resistant animal, you sort of upregulate.
So there are ontogenetic shifts in venom. So there's often a shift from more neurotoxic venom to a more hemotoxic and coagulopathic venom about the size at which snakes become adults and start to feed more on mammals and less on lizards. All of the snakes that I used here were adults, so we controlled for that ontogenetic shift in that way. There have been several studies of looking for seasonal variation or variation in response to stress, a whole slew of things, and basically most of it has turned up with a no. Once the venom is set, it doesn't have a lot of plasticity. There's expression variation, but it doesn't seem to respond to environmental cues, just growing up.
Do the snakes, I guess this is going off the plasticity thing, but do snakes, say, inject more venom if they are in a more resistant population, or do we have any idea about that?
We don't, although that would be a fascinating line of work to pursue as well. We do know that snakes inject more venom the study was done with this species at Loma Linda University down in LA. They inject more venom when the prey is larger. So there's sort of already a precedent there for snakes making those decisions, even though it's literally a split-second decision. But we don't know about choice of prey in terms of how resistant the prey might be. Fascinating direction. Yeah.
This is getting a little away from prey relationships, but for people developing antivenoms, does considering the population you're targeting possibly come into that?
Yeah, and that's one of the sources of or justifications for funding for some of the stuff that I'm doing now. We're working with folks in Brazil that are heavily involved in antivenom production to a lot of the big lancehead pitviper snakes that cause thousands of deaths and morbid injuries. The important thing is to use, and what's used now is a pool of different snakes. So we use, there are lots of different species that are pooled to produce antivenoms. But the more we can understand about, if we find a completely novel protein in one of the populations, you would want that in the antivenom to make it more effective. And the methods we're using now to understand which squirrel proteins are best at binding the snake proteins are actually derived from what's called antivenomics, which is how antivenoms are actually tested. Yeah.
So I have to ask the question, why are the higher elevation populations more effective with their venom? They have a larger effective population size?
We don't know about more effective. They just have higher metalloproteinase activity.
Oh, okay, I misunderstood.
They have higher protease activity in a sort of a gross sense of the measure. It's a main effect that's explained by elevation, not necessarily the interaction between snake and squirrel tissues.
Okay.
There's been a whole body of discussion on that as well. Are these proteolytic venoms helpful for digesting prey as an ectotherm when maybe the thermal environment isn't as friendly? You have colder nights, shorter active seasons. But there have been some respirometry type and other physiological approaches applied to this question that seem to suggest that no, venom isn't actually helpful for digestion. So
Isn't?
Is not. So
Generally, or that particular, or the
studies that have
metalloproteinase?
been done, they just injected venom or not, and then fed a dead animal to a snake, and digestive efficiency was not improved. So there's more to do there as well. I think it's something about some sort of trade-off, and maybe a trade-off in strategies between how these resistant squirrels are acting. The squirrels are less resistant in the places where the snakes have the most metalloproteinases, so it's a little bit counterintuitive in that regard. Yeah, again, can't give you any good answers, just thoughts.
In the populations where the squirrels are winning, are the snakes eating fewer squirrels?
We don't have any estimates of squirrel predation rates from these. I came in for 3 days to each of these sites, captured animals, took tissue, and left. I tried to get a surrogate for that with overlapping of niche models to see if maybe places that were highly suitable for both versus highly suitable for one, it didn't turn up anything, and so we need to, at some point, pick a few of these populations and actually do one of these intensive on-the-ground surveys of at least abundance.
What about a trade-off between toxicity and concentration?
There weren't concentration, so we
Of
did
the-
measure concentration of the
Right.
venom.
Right.
There,
That's what I'm saying.
there weren't any significant differences among sites in venom concentration. So it just seems to be about composition. Which metallic proteinases are present? How do those possibly interact synergistically with other toxins in the venom? These are these downstream questions that hopefully give me a career.
All right. Oh, yeah, go ahead.
Well, I was going to ask about the 25 metalloproteinase genes that, how divergent are they? One mechanism might be that there's just simple copy number variation that's really easy to evolve.
Yeah, there can be a lot. I can't give you any hard numbers, but there are quite a few SNPs present.
Are they tandemly duplicated, do you know?
There are some, I'm blanking on the word, but the little things that help duplications occur. It's, what?
Transposons.
Yes. There seem to be some of that involved. A lot of these are pseudogenized as well. So this fits the model of that you would expect under kind of an intense antagonistic coevolution where these duplicates are constantly being born and then dying when they maybe become useless pretty fast under strong selection. So we're gonna know more soon. The genome was just produced. I Skybeamed about it 3 days ago. That's when I learned this really
Okay, and Matt's going to be around for another 2 days, is that?
Yeah, at least until Friday evening. I'll be on the opposite corner, elbow deep in jars of snakes. So come find me, please.
All right, let's thank Matt
Thank you
once
all.
again.